Hello, I'm Duranch and welcome to the first episode of the Bionnovations podcast. Today we're going to have a little September recap where we're going to discuss maybe some recent scientific discoveries, some patents filed in the month of September. We'll be touching on what they are, how they could possibly be useful in the future. It's going to be split into two parts. The first part is just going to be sort of explaining the most recent scientific discoveries, whereas the second part is going to obviously be touching more on the patents. So yeah, September's been quite an interesting month, so we've got a lot to discuss and let's start. Okay, so to begin, late September gave us one of the more recent breakthroughs in Huntington's disease, introducing the first therapy which officially slows it down. I do also apologize in advance if I sound a bit robotic or monotone. I do have a script prepared and I am sort of going to go off the script a bit, so I do apologize. But for those of you who don't know, Huntington's disease has been the textbook definition of an untreatable genetic illness, being slow, irreversible, and causing the breakdown of brain function, being caused by a single mutated gene. Patients over time lose control of movement, memory, and personality over years, and until now, every available treatment has been to manage symptoms as opposed to actually trying to find a cure. However, in late September, research revealed something new, a new clinical gene therapy that appears to actually slow down the disease. The therapy currently being called Ep1, and in a 3-year trial, it reduced the rate of decline in motor and cognitive function by roughly 75%, which is not just an incremental improvement, it is actually the first real sign that we can change the course of the monogenic neurodegenerative disease. So let's unpack how this works. Ep1 uses an AAV vector, a harmless adeno-associated virus engineered to carry a small DNA sequence, which encodes a short hairpin RNA. Once inside the neurons, into critical brain regions, like the striatum, the RNA binds to the mutant HTT messenger RNA and basically tags it for removal, which lowers the production of the misfolding Huntingtin protein responsible for the neural damage. What's special here is its precision. It targets the root genetic cause without completely wiping out the normal Huntingtin function, which cells still need. Clinically, this change was dramatic. Participants who received the therapy maintained daily function, speech, and motor control for longer than expected, and a few even reported returning to work, something which was previously unheard of in the disease. And the surgery to deliver the therapy is only a one-time direct infusion into the brain. Is complex, but the safety profile so far is surprisingly manageable. From a biochemical angle, Ep1 is fascinating because it uses a molecular feedback approach, exploiting the RNA interface to rebalance the toxic protein network that otherwise spins out of control, and it's a kind of genetic precision control loop at the cellular level. From an innovation standpoint, it's obviously going to completely reshape the therapeutic strategy, not just for Huntington's disease, but for other protein misfolding disorders such as ALS and some form of Alzheimer's even. So we could probably expect a wave of patents around the viral delivery system, the specific RNA constructs, and even surgical targeting models, as companies obviously rush to adapt the framework to new diseases. So while Huntington's is not fully cured yet, this moment marks a turning point, being the first demonstration that a disease encoded in our DNA might be slowed and not just endured. Okay, so one of the next more exciting discoveries that September offered us was from Dr. Zhao Qingluo's lab at Purdue University, and his group basically uncovered a completely new type of ubiquitination process, one that bacteria use to hijack our cells, and it works in a totally unexpected way. So now, as most of us know, ubiquitination is a vital cellular process where small ubiquitin proteins are attached to other proteins and to mark them for degradation or to alter their function. It's a post-translational modification essential for maintaining cellular health. In human cells, this typically requires a whole enzyme cascade, E1, E2, and E3 enzymes that carefully transfer ubiquitin in a controlled manner. But Dr. Luo's work showed that some bacterial pathogens, such as Legionella pneumophilia, the bacterium behind Legionnaire's disease, don't actually play by these rules. Instead, they've evolved enzymes which can bypass the entire human ubiquitination process, and these bacterial enzymes, known as the SID-E family, use ADP ribosylation to attach ubiquitin to the host cell proteins. It's a biochemical shortcut which fuses together two key regulatory pathways, the NAD-based AD puribacillation system and ubiquitination, to manipulate the host cell more effectively. Essentially, these bacterial enzymes pull energy from NAD as opposed to ATP and perform E1 and E2 independent ubiquitination. This not only changes how the host signaling network behaves, but also allows bacteria to build a stable niche inside infected cells. Recent structural studies even showed how two of these effectors, LNA-B and MAV-L, coordinate through actin binding and AMPylation reactions to recycle modified ubiquitin molecules for reuse, a kind of biomedical recycling system. From a more molecular biology standpoint, these findings shakes up our understanding of post-translational modification. It proves that even core pathways like ubiquitination aren't unique to eukaryotes. They can be reimagined by pathogens for their own survival strategies, and it's a stunning example of evolutionary adaptation through enzyme innovation. For some of you more scientifically minded listeners, this also opens up new direction in infection biochemistry, where these alternate ubiquitination systems could be used as research tools to probe protein regulation, or even as synthetic biology components in the lab. And I think we could also expect some new patterns around these novel enzymes with their catalytic motives and their potential industrial or biomedical applications. Okay, so for our next September event, we've got a breakthrough which mixes cutting-edge engineering with core biochemistry, being a body-on-a-chip platform developed by researchers at Oxford. So this isn't just a catchy name, it's a miniature system where different types of living cells, think heart, liver, immune, and more, are grown in a tiny interconnected chamber on a single microfluidic device. So why is this so important for biochemistry? Because living systems are complex and cells never operate in isolation. And to truly understand how drugs or injury trigger immune responses, or how inflammation spreads and resolves across tissue, you need a setup which lets immune cells talk to their target organs the way they would in a real human body. The Oxford team's chip, released and validated in September, is especially exciting for heart research, as they've created a system where immune cells and cardiac tissue can interact dynamically after a simulated heart injury. This lets researchers watch in real time how immune cells migrate, what signals they release, and how those responses help or hinder tissue repair. For example, you can model a heart attack and then track exactly how neutrophils and macrophages coordinate cleanup and healing, or even see what happens when a drug is introduced to dampen the immune response. What's revolutionary here is the precision again. Rather than relying on animal models or oversimplified cell cultures, this chip allows for human-relevant experiments, mass data collection, and easy modification. One can swap in different cell types, genetic backgrounds, or drug regimens, and so it's like having a living, controllable, and scalable human biology model on your lab bench. From a biochemistry perspective, these platforms open enormous possibilities. You can study paracrine signaling, immune cell recruitment, and tissue immune crosstalk at unprecedented resolution, or test how new therapies in a human-like setting before moving to animal or clinical studies. And as a bonus, they may reduce animal testing and speed up drug development timelines. There's also a major space in the patent area too, as around this chip design, cell interface methods, and data integration algorithms. So yeah, body-on-a-chip tech represents a true convergence of biology, chemistry, and physics, and it might just redefine how we study disease and invent new medicines in the years ahead. Okay, so the next big story of September comes from the UC San Diego researchers, who unveiled an elegant advance in RNA-based editing, one which could make gene therapy both safer and more reversible. This study, led by Dr. Jean Yeo's lab, was published on September 18th and focuses on using small nuclear RNAs or snRNAs to precisely alter the RNA sequence of living cells. So, what's actually happening here? Many people have heard of CRISPR, the DNA editing tool which cuts and rewrites genetic codes permanently. RNA-based editing takes a different route. Instead of rewriting the genome itself, it makes changes at the messenger RNA level, temporarily altering how a gene is read without touching the original DNA. Think of it as making an edit to a working draft instead of scribbling on the master copy. Until now, RNA editors have struggled with efficiency and safety. They often caused off-target effects or fail to reach the right place of the cell. But this new approach uses small nuclear RNAs, native RNA molecules, which already live inside the nucleus as delivery scaffolds. The team fused these snRNAs with editing enzymes so they could swap out single RNA letters, like converting adenine to guanine or uracil to pseudouridine, directly inside the nucleus where RNA processing happens. This dramatically improved the precision and reduced mistakes. In test models, these snRNA-based editors corrected faulty RNA transcripts linked to cystic fibrosis, restoring normal protein production in affected cells. Unlike CRISPR, these edits were not permanent. Once the editor degraded, the cell's RNA returned to its original form. That makes the strategy both safer and more controllable, especially for conditions where temporary gene correction is preferable to irreversible change. From a biochemical view, this represents a fascinating convergence of synthetic RNA biology as well as natural cellular machinery. It's turning the nucleus into a precision biolab which can rewrite transcripts on demand. The enzyme at the heart of this process are often derived from ADARs, adenosine deaminases that convert A to inosine, which cells read as G. By engineering them to work out custom snRNAs, scientists have effectively built a programmable RNA repair toolkit. And the potential applications of this are huge, treating neurological, cardiovascular, and immune diseases caused by single-letter RNA defects safely and reversibly. And yes, patents have already been filed on both the engineering RNA scaffolds and the enzyme variants used for this process. For biochemistry lovers, this story is a perfect example of how far post-transcriptional biology has come. It's not about cutting DNA anymore. It's about fine-tuning its output. And as tools like these mature, they could redefine what gene therapy means, shifting us from a more permanent rewriting to elegant, reversible RNA tuning.
In terms of patent-related stuff, September also brought big stuff in terms of wearable diagnostics. Neutron makes a medtech startup from Melbourne, secured major funding and received pivotal patents for its lab-on-a-patch device, which is a platform that's changing how clinicians track and optimize therapies, especially for critically ill patients. So, it uses a set of DNA-based electrochemical sensors built into a macroneedle array, meaning dozens or even hundreds of biomarkers, proteins, metabolites, hormones, and even circulating drugs can be tracked continuously, painlessly, and on a single stick-on device. Unlike traditional wearables that only monitor glucose, this patch can also monitor molecules like lactate, vancomycin, which is an important but tricky to dose antibiotic, and kidney injury markers, giving doctors a much richer picture of what's happening inside the body minute by minute. ICU patients at risk for sepsis were given the lab-on-patch while receiving vancomycin, a narrow therapeutic window antibiotic that needs close blood monitoring to avoid kidney damage or underdosing. The patch let doctors see real-time drug levels and react immediately, improving safety and outcomes. From a biochemistry and engineering perspective, this is a giant leap, of course. The macroneedle technology samples the skin just deep enough to access interstitial fluid without pain or bleeding, and DNA aptamers, synthetic sequences, act as molecular locks for each analyte, enabling selective sensitive detection. And all of this data is pushed wirelessly to clinicians for rapid, precise therapeutic adjustment. Neutronic's September patent covers not just the hardware, but also the multi-biomarker analytics, cloud-based data streaming, and secure patient feedback. There's a real chance this tech could soon replace much of today's invasive blood sampling in critical care, outpatients, monitoring, and even chronic condition management. Okay, so to round off the more tech-heavy breakthroughs, let's look at the diagnostic side of biochemistry. So, how imaging and machine learning are making critical interpretations sharper, faster, and less error-prone. So, one of this month's standout IP developments came from GE Precision Healthcare, which secured a major patent for a new PET/CT motion correction system designed to eliminate the blur caused by respiratory and cardiac movement during scans. So, if you've ever seen a PET or CT scan in cardiology or oncology, you'll know how much the patient motion, even just breathing, can distort the final image. So small tumors and cardiac lesions can appear misshapen or even disappear depending on the movement during the scan. So GE's patent solution, building on its motion-free technology, takes real-time motion data directly from the scan itself, so no need for external sensors or chest belts, and integrates it live into the image reconstruction pipeline. Essentially, the system continuously tracks the position of organs using data-driven algorithms and corrects for motion artifacts frame by frame. Early evaluations show large improvement in signal-to-noise ratios and more accurate quantifications of tracer uptake, especially in cardiac scans where motion artifacts have traditionally made diagnostics tricky. From a more biochemical and clinical perspective, this directly enhances metabolic imaging resolution. PET imaging relies on radiochemical traces such as fluorodeoxyglucose to measure tissue metabolism, so improved correction means a clearer biochemical signature of what's really happening inside the body. Whether it's monitoring cancer metabolism or cardiac perfusion, it's a step towards more accurate forensics of biochemical processes in real time. September, in addition to this, also saw a new group of automated digital pathology patents rolled out, particularly those using AI for precise cell counting and classification on histology slides. These systems are convolutional neural networks to identify and count specific cell types in tissue samples, distinguishing normal from cancerous or inflammatory cells with high accuracy. And in test, AI-assisted systems reportedly match or exceed human pathologists in quantifying cells in complex structures, reducing error rates and processing times dramatically. Okay, so to finally round up September's biomedical patents, an invention which stands to make a real difference, especially in regions facing barriers to affordable healthcare. Early this month, Dr. Gayatri Gogoi, breast cancer researcher in Assam, India, was granted a design patent for a low-cost, eco-friendly breast self-examination model. The BSE device is crafted from basic sustainable materials like cotton cloth, wool, and lightweight plywood, making it safe, reusable, and low cost with a production price of just around 150 rupees, which is around £2. The tool is physically interactive, designed to help women and healthcare workers in rural and urban areas alike practice the correct self-examination technique by providing a hands-on model instead of relying only on diagrams or lectures. The invention boosts confidence, awareness, and the chance of detecting abnormalities early. So, why does this matter? In India, most breast cancer cases are still diagnosed late, largely due to a lack of awareness and limited access to mammography, especially in low-income and rural settings. Dr. Gogoi's model bridges this access gap. It empowers health workers to confidently demonstrate the process and trains women to identify warning signs themselves, all with tools that are portable and culturally not threatening. From a biochemistry and public health perspective, this patent is important for two reasons. First, it puts user-centered design at the heart of cancer screening, focusing on community education and empowering women. Second, it shows how innovation doesn't always mean cutting-edge electronics or AI. Sometimes using the right materials and human-centered thinking can have the greatest impact. So yeah, that's all for the September recap. I want to thank you all for listening, and I'll try to work on being a better presenter for next month's recap, but until then, have a good day.